Dear second semester students, so today I would like to explain cancer. Cancer is a uncontrolled cellular proliferation is called cancer. In this topic I am going to cover cancer classification, etiology, characteristics of the cancer cell, pathogens of cancer. So coming to the classification, two varieties of classifications are there. Coming to first one, based on nature of cell growth and invasion, cancer can be classified into two categories: benign neoplasm, malignant neoplasm. Here neo means new, plasm means cell growth or division. So neoplasm indicates new cell growth. So here benign neoplasm, in this we are going to observe unregulated cell growth without invasion or new growth will be observed. In malignant neoplasm, we will observe unregulated cell growth with invasion and metastasis. So metastasis means separation and migration of cancer cell into other places called metastasis. Penetration of such cancer cell into other organs is called invasion. Coming to the based on tissue of origin, from which tissue is participating in the cancer genesis. Based on that, three varieties are there. First variety is carcinomas. Carcinomas, the type of cancers are originated from the epithelial tissue. Sarcomas, these are originated from the non-epithelial tissues such as mesenchymal tissues. Leukemias or lymphomas, they are arising from the hemopoietic system and lymphoid cells. Coming to the etiology, there are so many causative factors are there. Among them, environmental lifestyle factors are most important for the genesis of cancer. So coming to first one, genetic factors are, that is family history. If the person is having a family history of cancer, the offspring is also getting chances of getting cancer. And obscure defensive system. So some races, inherently they are more tendency to get some varieties of the cancer. For example, Americans are more tendency to get breast cancer compared to the stomach cancer. Whereas Japanese, they are frequently encountering stomach cancer compared to the breast cancer. So next one is age. As the age increases, waste materials are deposited. There is a DNA damage takes place, which lead to the cancer. And radiation. So exposure to radiation, radioactive material also leads to the cancer. They directly damage the DNA, which lead to cancer. And next one is infections. Any infective material or virus which interacts with DNA, they leads to the cancer genesis, especially hepatitis B, C viruses, human papilloma virus, and Epstein-Barr viruses. And environmental factors and lifestyle factors such as smoking, food habits, consumption of street foods, and repeated cooking oils consumption leads to the cancer. And coming to the characteristics of the cancer cell will exhibit some of the peculiar characters to promote their cell development. So they will promote certain characters, they will inhibit certain characters. So these promotive factors, first factor is uncontrolled proliferation compared to the normal cell. And second variety is it is resistance to chemotherapy. It does not respond to the chemotherapy. And another character is migration. So these cancer cells will migrate, detach from the actual tissue or tumor and migrate to other place. That character is called migration. And this newly growing cell need more and more nutrition. That's why it will promote angiogenesis. More and more angiogenesis will be observed in a tumor. And next thing is, so these cells are resistant to die naturally. So that such process is called resistance to apoptosis. So these cells are also resistant to differentiation. Differentiation means the precursor cell may not convert into adult cells. That is called resistance to differentiation. And coming to the pathogenesis of cancer, there are four major pathways which are responsible for the pathogenesis of cancer. First one is reduced expression of AntiProliferation Gene, escape from the apoptosis and DNA repair. And next one is angiogenesis. Already I have shown angiogenesis. and increase telomerase expression. Coming to first one, reduced expression of anti-proliferative gene. Here CDK, cyclin dependent kinase inhibitor P21. P21 is an anti-proliferative gene which inhibits the CDK proteins. These CDK proteins promote the cell division. So this gene usually inhibits the CDK protein, so thereby inhibits the cell division. So in cancer cells, in many of the cancer cells, there is a reduced expression of such P21 protein is observed that leads to the reduced expression of anti-proliferative gene. And next one is escape from the apoptosis and DNA repair. So here apoptosis and DNA repair is controlled by the P53 gene. These P53 gene related proteins are usually stabilized in under oncogenic stress. If any stress is there, so these P53 proteins are aggregated to form a stable aggregate. This aggregate will promote P21 pathway to arrest the cell cycle, as I explained in previous slide. So this stabilized P53 also promote the apoptosis, nothing but programmed cell growth. Programmed cell death is called apoptosis. And next one is angiogenesis. So here angiogenesis is triggered by the cancer cell by hypoxia induced angiogenesis. Hypoxia encountered in a center of solid tumor. This promotes the release of hypoxia response genes. This leads to the release of hypoxia inducible factors alpha, HIF alpha, as well as vascular endothelial growth factors. These growth factors act on nearby blood vessels to promote new blood vessel formation. Same way, lymphoid vessels also, they will form a new lymphoid vessels. So usually these blood vessels will promote or carry the nutrients to the newly growing cell. Same way, the wastes are removed by the lymphoid vessels, which promote further cell division. And next thing is increased telomerase expression. Telomeres are the group of proteins which are present in the end part of the DNA. So usually during this DNA division, so here the nucleic acids base pair attachment is problem will be raised. That attachment problem will be resolved or solved by the telomerase enzyme. So in cancer cell, this telomerase expression increases. That indicates the cell is involved in the cancer. So spreading of cancer or metastasis, this is the additional information. So here this is the tumor cell. So after growing, the cellular junctions will be loosened. This lead to the metastasis. It will migrate to another place and it will invade into the a new area, that is lungs. And next one is some of the difference between the normal cell and cancer cell. So if you observe these cells, there are large number of dividing cells will be observed in cancer tissue, and a large variability shape of nuclei will be observed because there is a high number of DNA synthesis takes place. This lead to the large and variably shaped nuclei will be observed. Large nuclei to cytoplasm ratio. The ratio also changes will be also we are going to observe, and variation in size and shape. Normal cell having normal shape, but these cancer cell having variable in shape. There is no uniformity between these cells, and loss of normal cell features such as cell junctions, cellular extensions like axons. These are somewhat different from the normal cells. And next one is disorganized arrangement. The pattern of arrangement also different because the higher number of cells are there, their arrangement is not in a unique. And next variety is poorly defined tumor boundaries. So usually a normal cell having a certain normal boundaries, but in this case cancer cells there is no certainty of boundaries. So here some of the examples of the skin cancers. Moles also comes under the benign cancer. Thank you.